Good day, everyone. Today we will be learning the first unit of the evolution laboratory, which is called Population Genetics and Evolution. First, let's have a recap on some key terminologies. Population genetics is the study of genetic variation within and among populations and the evolutionary factors that explain this variation. But what is a population? A population is a group of organisms belonging to the same species that are found in the same area and can interbreed. It is the smallest unit that can evolve, and this means that an individual cannot evolve. A population also has this so-called gene pool. A gene pool is a combined genetic information of all the members of a population. Both populations and gene pools are dynamic, meaning they can change over time and space. In this unit, we will be investigating these agents of change in the gene pool of a population, which are also referred to as evolutionary forces. Major evolutionary forces include, first, mutation, which is the ultimate source of genetic variation, and it is the initial requirement for evolution to occur. Next is the gene flow or the migration, which is the movement of genetic information from one population to another. Genetic drift, which is the change in allele frequencies merely due to chance, and it is random. Lastly, natural selection, which is the survival of the fittest, where the most advantageous traits for survival and reproduction are selected. This exercise focuses on gene flow, genetic drift, and natural selection, and how they can change the gene pool, or more specifically, the allele frequencies of a population. Hardy-Weinberg equilibrium will be employed as a reference model to measure if these forces are acting on a population. It serves as a reference or baseline because it describes the hypothetical, non-evolving populations. Hardy-Weinberg principle have 5 conditions, which include no mutation, no migration, no selection, population is large, and mating is random. However, natural populations hardly meet all the 5 conditions, and when the conditions are not met, the difference between the actual gene frequencies and those expected under the ideal Hardy-Weinberg conditions can be determined. This difference is the measure or the rate of evolutionary change. Let us assume we are studying a population of a frog with members that have a dark green and light green colors. The dark green frog carries at least one dominant allele, big G, while the light green color carries 2 recessive alleles, small g. It is also given that the frequency of the dark green allele is 0.6 and the frequency of the light green allele is 0.4. The population under consideration is in Hardy-Weinberg equilibrium, and the big G and small g alleles are not carried in the same sex chromosomes. Under these conditions, any offspring in the population has a 0.6 or 60% chance of receiving the dark green big G allele from either parent and a 0.4 or 40% chance of receiving the light green small g allele. Recall that we are dealing with an entire population, and these values represent average probabilities across the entire population. Now let's use this information to draw out Hardy-Weinberg formulas. First, let us now consider a single genetic locus with 2 alleles, big G and small g, and let P as the frequency of occurrence of the big G allele and Q as the frequency of occurrence of the small g allele. Notice in the example that the percentages add up to 100% and the frequencies add up to 1. The first formula of the Hardy-Weinberg equilibrium therefore is P plus Q equals 1. Given the P and Q allele frequencies, the Hardy-Weinberg equilibrium allows us to calculate the genotype frequencies in the population as follows, where P squared is the frequency of the homozygous dominant, 2PQ is the frequency of the heterozygous genotype, and Q squared is the frequency of the homozygous recessive. Now let's input the P and Q values in the formula. Therefore, the overall probability for each expected genotype in this population is shown as follows. We are done with the background. Let us proceed to the actual exercise.
Let us first identify the materials needed for the experiment. "500 pieces of small-sized beads in colors pink, white, red, and green. 200 pieces of medium beads in colors red, pink, and white. 4 containers or shoe boxes and 3 Ziploc bags. 1 makeshift sieve. The sieve should be enough to allow only the small and medium-sized beads to pass. And finally, 1 pair of forceps. Let's proceed to the general procedure. Place all of the small-sized beads of a single color into one of the 4 shoe boxes. Label the box with the pink beads East, the box with the red beads North, the box with the white beads South, and the box with the green beads West. Place the 4 boxes with beads along the 4 sides of your island. Place the medium-sized beads into 3 small Ziploc bags. You should put about 200 beads of a single color into each bag. In this exercise, you will be working with a population of gravel divers. Gravel divers, which look very much like medium-sized beads, can exist in one of the 3 different colors. The red gravel divers are homozygous for the capital A allele. The pink gravel divers are heterozygous for the 2 alleles. The white gravel divers are homozygous for the small letter A allele. Gravel divers normally bury themselves in gravel, which looks very much like small-sized beads. On the exotic islands where these gravel divers are found, the color of the gravel varies according to geographical location. Specifically, on the north side of the island, the gravel is red. On the east side of the island, the gravel is pink. On the south side of the island, the gravel is white. And on the west side of the island, the gravel is green. The native predator of the gravel diver is an organism called the gravel diver picker. This organism will be represented by all or any members of the lab group. Now, let's proceed to study the gene pool of the gravel diver population and the impacts of various evolutionary forces on this gene pool. The procedure has 4 subparts. First, the gene pool. Second, the gene flow. Third, the natural selection. And lastly, the random genetic drift. Let us go to the first part, the gene pool. Remember that there are 4 different study sites: north, south, east, and west geographical locations being researched. The initial frequency of the red, pink, and white gravel divers varies according to the specific geographical location on the island. In the north gravel location, which is the red gravel, add 64 red, 32 pink, and 4 white gravel divers. Gravel divers like to burrow in the gravel, so be sure to bury your gravel divers in the gravel thoroughly. In the east, add 49 red, 42 pink, and 9 white gravel divers. In the south, add 36 red, 48 pink, and 16 white gravel divers. In the west, add 25 red, 50 pink, and 25 white gravel divers. Record the count of each color in each gravel location. Calculate the genetic and allele frequencies of each gravel diver population per geographic gravel location. Remember that the color alleles of gravel divers are codominant. In other words, each red gravel diver carries 2 red alleles for capital A. Each pink gravel diver carries 1 red with a big letter A and 1 white for the small letter A allele. Each white gravel diver carries 2 white alleles for small letter A. Therefore, we can directly divide the number of gravel divers in each color by their total number. The frequency of red gravel divers is equal to the number of red gravel divers over the total number of gravel divers. The frequency of pink gravel divers is equal to the number of pink gravel divers over the total number of gravel divers. The frequency of white gravel divers is equal to the number of white gravel divers over the total number of gravel divers. With the calculated genotype and phenotype frequencies, the frequency of the red capital A allele in the gravel diver population can now be calculated using this formula. P represents the red allele frequency in the gravel diver population is equal to 2 multiplied by the total number of red gravel divers in the population plus the total number of pink gravel divers in the population over 2 multiplied by the total number of all gravel divers in the population. And with a calculated p-value, we can utilize this formula to calculate for q, which is p plus q is equal to 1. Using this equation, we can transpose p to the other side, giving us q is equal to 1 minus p, where q is equal to the white allele frequency in the gravel diver population. These calculated frequencies are the initial gene pool frequencies of your gravel diver population. Tabulate your calculations in the table provided in the manual. Use the P and Q values that you entered above to calculate the expected genotype frequencies given the assumptions of the Hardy-Weinberg principle. Compare your expected frequencies to the observed frequencies that you entered in Table 1.1. Is your population in Hardy-Weinberg equilibrium? Why or why not?
In the next sections, we will be investigating if there will be changes in the initial genetic and allele frequencies when an evolutionary force is acting on it. Next, let's go to gene flow. Spring has come to your island, and the reproductively mature gravel divers are anxious to find a mate. The gravel divers at the 4 geographical locations on your island tend to behave somewhat differently during mating season. The gravel divers living on the north and south sides of the island are basically happy homebodies. They like the other gravel divers in their own population, and they just find a neighborhood male or female with which they are compatible and mate. The gravel divers living on the east and the west side of the island are more adventurous. Many of these gravel divers commonly leave their natal population to find a mate. Some of the gravel divers from the eastern shore will migrate to the southern shore, and some of the gravel divers from the western shore will migrate to the northern shore. The objective of this part of the experiment is to determine the impact of gene flow on the populations. First, let's generate a hypothesis. What do you expect to happen at the study sites? For each of the following genotype and allele frequencies, clearly indicate whether you expect the frequency to increase, decrease, or remain the same after migration has occurred. After writing your hypothesis, let's now proceed to the experiment. If the population is on the east side, some of your gravel divers will migrate to the south. We will use a sieve to simulate the migration. First, the eastern population will be sifted once using a sieve. After, count the sifted gravel divers of each color. There are 4 red, 6 pink, and 4 white gravel divers. Take note and record the result, and after, transfer all the sifted gravel divers to the southern island. Repeat the procedure, but this time, the movement is from west to north. Perform calculations using a similar method in the previous section to determine the new allele and genotype frequencies after gene flow. Write all your results on the table given in the manual. Answer the questions and analyze the effect of gene flow to the gravel diver population by comparing the expected and observed genotype frequencies. To have a clear perspective on the experiment, let's now take a look at this video. The next evolutionary force is natural selection. Here, we assume that after migration, the survival of the gravel divers is now primarily affected by the predation of the gravel diver picker, or GDP. As we have previously noted, the GDP looks very much like you and your lab partner. Generate first a hypothesis whether you expect the frequency to increase, decrease, or remain the same after natural selection has occurred. After writing your hypothesis, let's now proceed to the experiment. Choose one person in your lab group to be the predator. This person must use a pair of forceps when preying upon the gravel divers. The use of forceps simulates the time it takes a GDP to find and immobilize a gravel diver. Choose a second person in your lab group to be the timekeeper. We will start in the north. Mix the gravel divers first. After, the GDP will capture one gravel diver at a time with the use of forceps in a span of 1 minute. count and record the captured gravel divers. We have 1 red, 4 pink, and 3 white gravel divers. Repeat the procedure for the east, west, and south sides. After, calculate the new allele and genotype frequencies after natural selection by using a similar method in the previous section. Write all your results in the table given in the manual. Answer the questions and analyze the effect of natural selection to the gravel diver population by comparing the expected and observed genotype frequencies. To have a clear perspective on the experiment, let's now take a look at this video. The last part of the experiment is random genetic drift. The scenario here is that natives went to the gravel locations at night and randomly collected 6 gravel divers and brought them to their island. These gravel divers are now the founding population in the new island. We then assume that the small gravel diver population continues to survive and reproduce in the new island. We are now simulating a specific example of genetic drift called the founder effect. Generate first a hypothesis whether you expect the frequency to increase, decrease, or remain the same after genetic drift has occurred. After writing your hypothesis, let's now proceed to the experiment. In each of the geographic locations, a random sample of gravel divers will be taken from the population with the use of a sieve. From the sieved samples, 6 gravel divers will be randomly picked by a member with his or her eyes closed. These 6 individuals represent the founders that will be brought to the new island and start the new population. Repeat the procedure for north, east, and south sides. After you're done with the experiment, perform calculations using a similar method in the previous sections to determine the new allele and genotype frequencies after random genetic drift has occurred. Answer the questions and analyze the effect of random genetic drift. To have a better grasp of this part of the experiment, let us now take a look at this video. That concludes the entire presentation. Thank you for staying this far, and we hope that you are now equipped with the basics needed for this laboratory exercise. The complete and more detailed instructions are included in the laboratory manual, so please read them as well. Also, don't forget to incorporate all the guide questions in your laboratory report and follow the proper format.